Molecular transport refers to the movement of compounds across the outer wall or membrane of the cell. This transport is critical in two respects. Firstly, transport allows the cell to take in and release compounds in accordance with its biological function. For example, the uptake and release of oxygen by red blood cells. Secondly, it allows cells to regulate characteristics of this transport. With this presentation, you will gain a better understanding of the different types of transport that occur across a membrane barrier. Every cell is surrounded by a plasma membrane that separates it from the extracellular environment. The plasma membrane serves as a permeability barrier that allows the cell to maintain an interior composition far different from the composition of the extracellular fluid. The basic structure of the cell membrane is a lipid bilayer which is composed entirely of phospholipids and cholesterol. A phospholipid molecule consists of a polar head group and two nonpolar hydrophobic fatty acyl chains. One part of the phospholipid and the cholesterol molecule is soluble in water, that is, hydrophilic, whereas the other part is soluble only in fats, that is, hydrophobic. Because the hydrophobic portions of both these molecules are repelled by water but are mutually attracted to each other, they have a natural tendency to line up, occupying the center of the membrane, and the hydrophilic portions projecting to the two surfaces in contact with the surrounding water. Compartmentalization, or maintenance of differences in fluid composition across the membrane, is an important way wherein cells regulate their own activity. This is achieved because of the properties of the compartment's barriers. Molecules are in a continuous state of movement. The warmer the environment, the more rapid is the movement. The movement is known as the random thermal motion. The movement of molecules from one location to another due to random thermal motion is known as diffusion. This proceeds from an area of higher concentration to an area of lower concentration. Ions diffuse across the membrane via the integral proteins known as channels. The selectivity of channels is dependent on its diameter and electrical charge. They are distinguished by two important characteristics. Firstly, they are often selectively permeable to certain substances. And secondly, many of the channels can be opened or closed by gates. The process of opening or closing of the ion channel is known as channel gating. Ion channels can be classified according to the process that will change their conformation. Firstly, we have the ligand-gated channel. This ion channel opens when a specific molecule, called a ligand, binds to it. We also have the voltage-gated channel. This channel type opens when a change in membrane potential occurs. Some substances enter or leave cells via the integral proteins of the plasma membrane, called transporters or carriers. A transporter binds the solute to be transported on one side of the membrane, and then the transported undergoes a conformational change that allows the solute to be released on the other side of the membrane. There are two types of mediated transport system: facilitated diffusion, sometimes called facilitated transport, and active transport. Facilitated diffusion occurs via a transporter that does not require an input of energy and are therefore not capable of transporting molecules against its concentration gradient. The movement proceeds from an area of higher concentration to an area of lower concentration. Active transport is the pumping of molecules against their concentration gradient with the expenditure of energy in the form of ATP. The movement of molecules proceeds from an area of lower concentration to an area of higher concentration. Active transport is divided into two types according to the source of energy used to cause the transport. These are called primary active transport and secondary active transport. Primary active transport directly uses ATP to transport solutes. The transporter is usually an ATPase enzyme that breaks down ATP. The phosphorylated transporter undergoes conformational changes, which helps transport substances across the membrane. A good example is a sodium-potassium ATPase pump. It pumps 3 sodium ions out and 2 potassium ions into the cell. On the other hand, secondary active transport uses the stored energy of an electrochemical gradient to move both an ion and a second solute across the plasma membrane. The ion moves along the electrochemical gradient, while the second solute moves against its concentration gradient. If the solute moves in the same direction as that of the ion, the process is called symport or co-transport. An example of this is a sodium-glucose exchanger, where glucose moves against its concentration gradient and in the same direction as that of the sodium ion. If the solute moves in the opposite direction as that of the ion, the process is called antiport or countertransport. An example of this is a sodium-calcium exchanger. It uses the electrochemical gradient of sodium to pump calcium out of the cell. 3 sodium ions enter the cell for each calcium ion that is extruded. There is another way by which substances can enter or leave the cells without passing through the structural matrix of the plasma membrane. These processes are called endocytosis and exocytosis. Endocytosis is a process whereby the plasma membrane encloses an extracellular material, folds into the cell, forming small pockets that pinch off to produce the intracellular vesicles. The uptake of articulate material is termed phagocytosis, while the uptake of soluble materials is called pinocytosis. Exocytosis is the process whereby the membrane of an intracellular vesicle fuses with the plasma membrane and subsequent expulsion of its content. The traffic of molecules through biological membranes is vital for many cellular processes. The different transport systems enable our system to absorb nutrients needed by our body for its vital functions. An example is glucose absorption. It occurs in a co-transport mode with the active transport of sodium. The initial active transport of sodium through the basolateral membranes of intestinal cells provide the force for moving glucose through the same membrane. Transport proteins in the brush borders will not transport sodium into the cell until sodium combines with glucose. On the other hand, glucose exits the epithelial cells through the basolateral membranes by way of facilitated diffusion.
Presented is a comparison of the pathways by which substances cross the membranes. For the direction of net flux, diffusion either through a lipid bilayer or through a protein channel, as well as facilitated diffusion, is from an area of higher concentration to an area of lower concentration, while primary and secondary active transport is from an area of lower concentration to a higher concentration. For the energy requirement, diffusion through a lipid bilayer or through a protein channel, as well as facilitated diffusion, do not use energy, while primary and secondary active transport use energy in the form of ATP. For the use of integral membrane proteins, diffusion through a lipid bilayer does not use an integral membrane protein, while diffusion through protein channels utilize integral membrane proteins. For mediated transport pathways, facilitated diffusion, primary active transport, and secondary active transport all make use of integral membrane proteins in the form of carriers.